I'm Addison Randolph. Today I would like to discuss controversial debate sparking gene therapy and whether or not it should be used to cure various human diseases. Gene therapy. Is it ethical? Is it worth the risk? Will it really work? These are all questions that spark debate amongst many. But before I present the various opinions on why or why not gene therapy should be used as a form of treating diseases, I would like to inform you what exactly gene therapy is. According to the Pearson Baccalaureate Higher Level Biology textbook, gene therapy is a treatment strategy which involves the introduction of genes into human cells to alleviate disease. In order to try to accomplish this, defective genes are replaced by effective genes, which send off the message to make the correct proteins. These effective genes are delivered to the appropriate cells by vectors, or viruses which carry new DNA with the correct genes and have been genetically engineered to infect the appropriate cells. There are two types of gene therapy: germline therapy and somatic therapy. Germline therapy targets germline cells, or egg and sperm cells. This results in the passing down of any genetic changes to offspring and following generations of that patient. Unlike germline therapy, somatic therapy targets somatic cells, or the majority of other cells in the body. This form of gene therapy only allows for the genetic changes to take place in the patient, meaning that the changes will not pass on into the offspring and following generations of that patient. There are two methods of performing somatic gene therapy: ex vivo and in vivo somatic therapy. Ex vivo or external somatic therapy modifies cells outside of the body. Cells from the bone marrow or the blood of the patient are extracted and exposed to the virus with the correct gene. They are then injected back into the patient's bloodstream. In vivo or internal somatic therapy transfers the viruses containing the correct gene inside the patient's body. This is the more popular form of somatic gene therapy, since it is much less painful for patients than ex vivo somatic therapy. In order to further explain what happens during in vivo somatic therapy, I would like to present a short clip from Cortical Studios. This shows vectors entering into the bloodstream in order to reach cells within the liver.
Virus particles containing the therapeutic gene enter the liver with the bloodstream. Their target is the region where liver cells exchange matter with the small capillaries. There are large pores in the capillary walls. The liver cells are on the other side of these openings. The capillaries also contain special cells, the macrophages. Their task is to remove any foreign and harmful particles. They will try to do this also with the virus particles. That is why the outside of our virus is coated with a protein that can bind specifically to liver cells. Because of this protein, the virus will get past the macrophages and reach the liver cells more quickly. Once the virus particle binds to the liver cells, the virus's envelope merges with the liver cell's cell membrane. This allows the virus's load of therapeutic DNA to penetrate the cell. It will find a path to the cell's nucleus. The virus proteins assure that the therapeutic gene is taken into the genetic material of the liver cell. This will enable the liver cell to function once again as a healthy cell.
Now that I have informed you what gene therapy is, in general terms, I would like to present you with the risks and benefits of it, as well as some arguments as to why gene therapy should or should not be used. According to the Pearson Baccalaureate Higher Level Biology textbook, the risk of gene therapy include, as listed on the slide, The virus vector used to deliver the gene may enter the wrong cells. The virus vector may cause a mutation by placing the new gene in the wrong location of the chromosome. Genes can overexpress, creating too much protein. The virus vector may stimulate an immune reaction. The virus vector might be transferred to others like cold or flu viruses. And children may be more sensitive to long-term hazards since their tissues are still developing. Benefits of gene therapy according to this textbook include the possibility of curing a disease caused by a single gene defect and the possibility of curing a disease caused by multiple genes as defects. As you can see, the number of risks possible when using gene therapy is significantly greater than that of the number of benefits. Some view these risks as an argument not to use the method for curing diseases and genetic disorders. Others have also created argument that this method is unethical because of religious and other personal beliefs, such as the Vatican of the Roman Catholic Church. His argument will be presented in this video clip.
The dignity of a person must be recognized in every human being from conception. This fundamental principle expresses a great yes to human life and must be at the center of ethical reflection on biomedical research. That's the message of Dignitas Personae, a new document on biomedical research ethics published by the Congregation for the Doctrine of the Faith. It's an update of Donum Vitae, the Congregation's document on the same subject published in 1987. The Vatican says this document is still relevant, but needed to be revised because of new developments in the field of science during the last 21 years.
Le nuove tecnologie da una parte. On the one hand, new technologies open doors for new potential treatments that were previously unknown, such as infertility treatments and the use of adult stem cells. On the other hand, these raise serious questions on the anthropological and ethical nature.
The document acts as a guideline for Catholics in and out of the field of science. It outlines the Vatican's opposition to research using stem cells taken from living human embryos, the morning after pill, gene therapy intended to affect a patient's offspring, embryo manipulation, freezing of human embryos or eggs, and cloning.
Ciò che questa istruzione intende fare. What this document intends to do is express its own authoritative contribution to the formation of the conscience of not only believers, but of so many who want to hear the arguments of the Church and face them.
Theological and ethical controversies over scientific research are not going away anytime soon, but the goal of this document is to give doctrinal guidance to Catholic patients and scientists on what procedures the Church does and does not approve of based on ethical and moral criteria.
Gli argomenti non sono frutto del... Arguments are not the product of faith. Arguments are the product of reason. I want to repeat that the document speaks more of ethics, especially when there are these arguments, because ethics is correctly the expression of reason, which during its search of how to direct life, it does so under the light of reason.
Dignitas Personae draws from a variety of sources, including its predecessor Donum Vitae, John Paul II's 1995 encyclical Evangelium Vitae, and the teachings of Benedict XVI. It makes note of his address on stem cell research to the Pontifical Academy of Life in 2006. The document, which was published with Benedict XVI's approval, calls on Catholics to courageously oppose injustices against the dignity of human beings, including the unborn.
The argument for the use of gene therapy that I would like to present to you is in the form of an analogy taken from the University of Utah. Imagine that you accidentally broke one of your neighbor's windows. What would you do? You could stay silent. No one will ever find out that you are guilty, but the window doesn't get fixed. Or you could try to repair the cracked window with some tape. Not the best long-term solution, however. Or you could put in a new window. Not only do you solve the problem, but also you do the honorable thing. So, if a flawed gene caused our broken window, can you fix it? What are your options? You can stay silent, ignore the genetic disorder, and nothing gets fixed. Or you could try to treat the disorder with drugs or other approaches. Depending on the disorder, treatment may or may not be a good long-term solution. Lastly, you can put in a normal functioning copy of the gene. If you can do this, it may solve the problem. This analogy appears to be arguing that gene therapy is the most honorable or overall best way to fix a genetic disorder. Therefore, it is arguing for the use of gene therapy. As you can see, there are extreme arguments for and against gene therapy. I would like to show you a point of view on a topic from a student. The student in the video makes a very good point about how medical developments occur, encouraging further research of gene therapy use to take place. She does address an argument, however, against gene therapy use in terms of its ethical implications at the conclusion of the clip. Overall, though, she does do a good job of addressing both sides of the argument. So here's the clip.
I'm not sure about all this genetic engineering and gene therapy stuff. I'm not sure if it should be allowed or not. I think it's worth trying out new treatments if it means that it could help people in the future, especially if it means that people get to live longer or get a better quality of life. But I still think there is so much more research needs to be done before scientists can be sure that it's completely safe for humans. But that's how medicine progresses, by trying out new treatments, even if the early patients die. I mean, that's what happened with heart transplants, and look how many lives they save now. I'm still not completely convinced. I mean, gene therapy is manipulating genes, and that's a bit like interfering with nature, and I'm not sure that that's the right thing to do.
I still have not answered the big question. Should gene therapy be used to cure human diseases and other genetic disorders? To help us reach a conclusion, I would like to show you another clip. This does not demonstrate the curing of a disease with the use of genetic therapy, but it does show the successful betterment of eyesight for blind patients whose blindness was caused by a genetic disorder with the use of gene therapy.
A light at the end of the tunnel may soon appear for those suffering a form of genetic blindness. University of Florida researchers have discovered that retinal gene therapy can improve the eyesight of people born with a gene defect that robs them of their vision.
The gene essentially goes into the cells that is missing it, and it starts expressing the protein that it encodes, and that protein is the enzyme they're missing. So this enzyme then restores the visual cycle to these cells.
UF researchers injected the gene into the eyes of 3 young adults with a type of blindness called LCA2. They found the patient's eyes were 10,000 times more functional in the region into which the therapy was injected. One woman could see a digital clock for the first time in her life.
Her brain had realized that we had restored some function to a non-central part of her retina, and her brain had now rewired the muscles of her eye so that depending on the brightness of the object, she would use either the central retina or the treated part of the retina.
People with sight have one central vision or fovea they can see out of, but this gene therapy created another off-center region that has become more sensitive to light. At the University of Florida, I'm Quentin Iman.
Another more recent success story has been published. According to the Associated Press and researchers from the University of Pennsylvania, gene therapy has successfully treated leukemia by turning patients' own blood cells into assassins that hunt down and wipe out their cancer. Only 3 patients were tested, 2 of which who have been cancer-free for the last year and 1 of which has had a partial response. For the experiment, blood was taken from each patient and T cells removed. After they were altered in a lab, millions of these now T cell attack viruses were returned to the patient in 3 infusions. These cells not only work to kill the existing cancer cells, but also kill new cancer cells as they emerge. Some side effects, such as severe nausea, fever, and other flu-like symptoms, were felt by the patient. Further studies will be done to see if these effects can be reduced. Studies to use this technique to fight other cancers are already being performed, and according to Dr. Jennifer Ashton, this is the wave of the future. Therefore, I have come to the conclusion that gene therapy is something that should continue to be tested and tried on various patients who have no other hope. I feel this treatment will further progress until it is deemed safe as a treatment to cure multiple diseases and other genetic disorders. So, should gene therapy eventually be used to cure diseases and other genetic disorders? Yes, I think it should.